So welcome back, my dear students. Today I shall be continuing with the further discussions on the physiology of hearing. In my last class I have stopped at the functional anatomy part of the cochlea of the inner ear. I haven't got into the depth of the functional anatomy. Today I shall be discussing the organ of corti or corti and the hair cells and their functional anatomy in details. Thereafter I shall be taking up the topic of sound transduction. So let's begin our discussion for today. Firstly, the organ of corti or corti. Where is it located? It is located on the basilar membrane. If you remember, the basilar membrane is the basal membrane of the membranous labyrinth. On top of that membrane is located the organ of corti. Now this organ of corti is important because it is the structure which holds the hair cells. As I have said, the hair cells are the auditory receptors. So the organ of corti is the most important organ for hearing of the inner ear. Now how is it disposed? Just like the cochlea, the organ of corti extends from the base till the apex of the membranous labyrinth. So basically it is also coiled like the cochlea, and hence it's also sometimes called the spiral organ of corti. On top of that is located the hair cells on top of the organ of corti. This organ of corti has a tough reticular membrane-like lamina. The hair cells which are embedded in the organ have their tips, or rather their tops, jutting out from the organ of corti, and they pierce the tough reticular-like lamina, and when they pierce this tough reticular-like lamina, there are certain cells called pillar cells which support these hair cells. These pillar cells are also sometimes known as the rods of corti. Now, within the organ of corti, the hair cells are arranged in 4 rows, but these 4 rows are not homogeneous. Actually, there is a structure known as the tunnel of corti formed by the pillar cells or the rods of corti. 3 rows of cells are arranged lateral to the tunnel, and a single row is arranged medial to the tunnel. Those which are arranged lateral to the tunnel are known as the outer hair cells, and the row which is medial to the tunnel is known as the inner hair cell row. There are about 20,000 outer hair cells and 3,500 inner hair cells in each human cochlea in an adult. Now, these rows of hair cells, as I have already said, are jutting out from the top of the organ of corti. Now, covering the rows of these hair cells, or that is on top of the hair cells which are jutting out, there is a thin but viscous and elastic membrane known as the tectorial membrane. The tips of the hair cells, of the outer hair cells, are embedded in this tectorial membrane. So basically the outer hair cell tips are embedded in the tectorial membrane above, and the hair cell itself is embedded in the organ of corti below. But for the inner hair cells, the tips are not embedded in the tectorial membrane, they are free. This is a schematic diagram of the organ of Cautai on the left hand side. You can see it shows the three scali or scala vestibuli, scala media and scala tympani. You can see the basilar membrane has been pointed out, and on top of it is the organ of Cautai. The pillar cells forming the tunnel has been shown in red, while the hair cells has been shown in blue. Here you can see the three layers or three rows of outer hair cells and a single row of inner hair cell is shown. The tectorial membrane is shown. Also important thing which is shown is the location of the spiral ganglion, which is the ganglion of the vestibulocochlear nerve, and another structure which is shown on the wall of the scala media, which is given the name of stria vascularis. This is the structure from which the endolymph is secreted. The right hand side is a much more enlarged photograph of the organ of Cautai. You can see the inner hair cell, you can see the outer hair cells. You can see in the picture the outer hair cell tips are embedded in the tectorial membrane, while the inner hair cell tip and the tectorial membrane has a small gap. In the picture also it is shown. Other cells and few other nomenclatures you can see in the diagram. They are basically part of histology, and you will learn about them in details in your anatomy histology class. Just try to appreciate the fact that the inner hair cells and the outer hair cells are arranged in rows with their tips jutting outwards from the organ of Cautai. They are bathed in endolymph. The tip of the inner hair cell is free, while the tips of the outer hair cells are embedded in the tectorial membrane. The hair cells, being the receptors of sound, needs to transmit whatever they are receiving to the nerves for the brain to perceive it as sound. So what is the arrangement? In the preceding diagrams I have shown you the location of the spiral ganglion. The spiral ganglion is the collection of cell bodies of sensory neurons, and the axons of these cell bodies arborize around the bases of the hair cells. The spiral ganglion, as shown in the picture, was located within the bony core of the cochlea, that is the bony labyrinth. That bony core also has a name. It is known as the modiolus. So again, the cell bodies of the ganglion that is responsible for the first reception of signals from the hair cells is located within the modiolus of the cochlea, and it is known as the spiral ganglion. It has been seen that 90 to 95% of the sensory neurons from the spiral ganglion innervate the inner hair cells, and only 5 to 10% innervate the outer hair cells. So the inner hair cells are much more important as receptors for sound compared to the outer hair cells. Now the axons of the afferent neurons that innervate the hair cells, they form the cochlear division of the 8th cranial nerve. So the ganglion is the spiral ganglion. From it, one process reaches the hair cells, while the other process, which are going towards the central nervous system, they all club together to form the cochlear division of the 8th cranial nerve. Other than the afferent innervation that the outer hair cells receive from the spiral ganglion, it has been observed that they also receive cholinergic innervation via an efferent component of the auditory nerve. Efferent and afferent concept needs to be cleared at this junction. Afferent means any nerve that moves towards the central nervous system, and efferent is any nerve that comes from the central nervous system towards effector organ. The sensory nerves of the spiral ganglion are afferents, that is, they receive signal from the hair cells and transmit them towards the central nervous system. But the outer hair cells, which are basically receptors, receive an efferent cholinergic innervation from the auditory nerve. The physiological significance of such innervation is still a bit unsettled, but it has been postulated that this innervation is responsible for the control that the outer hair cells have over the inner hair cells regarding the latter's sensitivity to sound. Means the main receptors for sound are the inner hair cells. They receive the lion's share of the sensory innervation. The outer hair cells are not much responsible for sound perception. They have other role. And what is that role? They control the sensitivity of the inner hair cells to sound, and they do so through the cholinergic innervation they receive from the auditory nerve. This is a postulate. I am telling you again, it is still not settled, but it is almost confirmed these days. It is believed that the cholinergic innervation leads to hyperpolarization of the outer hair cells, which alters their length and stiffness, resulting in tuning of the auditory sensitivity of the inner cells. Or the outer hair cells become stiff and their lengths alter due to a hyperpolarization caused by the cholinergic innervation, and this stiffness and alteration of length results in tuning of the auditory sensitivity of the inner hair cells. Another functionally important anatomical structure within the cochlea is the presence of tight junctions between the hair cells and the adjacent phalangeal cells. This presence of tight junction prevents endolymph from reaching the bases of the cells. But the basilar membrane, which is forming the base of the organ of Corti, is relatively permeable to the perilymph in the scala tympani, and consequently the tunnel of the organ of Corti, which is formed by the pillar cells, and the bases of the hair cells are bathed in perilymph, while the hair cells top and the tips are bathed in endolymph. So this is one anatomically important consideration which will affect its physiological role. As I proceed my lecture, you will understand the importance of this. Now let's discuss the hair cells in slight details. As I have already discussed, this organ of Corti is essentially a narrow sheet of columnar epithelium cells which contains the sensory hair cells and their accompanying supporting cells. Now each of the 3500 inner hair cells are innervated by about 10 axons of large bipolar cells in the spiral ganglia. Now again, let's recap. The spiral ganglion is the sensory ganglion from which the processes arborize at the bases of the hair cells. 90 to 95% is arborizing at the bases of the inner hair cells, while only 5 to 10% on the basis of the outer hair cells. Now, the types of cells in the spiral ganglia also vary. There are two varieties. One are the large bipolar type 1 cells, the others are the type 2 cells. The type 1 cells, they innervate the 3500 inner hair cells. And what is the nature of such innervation? Each inner hair cell receives about 10 axons of large bipolar cells. So one hair cell is receiving 10 axons. From my class of synapse, you can recall that this is an example of divergence. Means one receptor is giving outputs to 10 neurons. So that is a case of divergence. The 20,000 outer hair cells are innervated by axons from small bipolar or type 2 cells in the spiral ganglia. Each of these synapses with about 10 outer hair cells. Thus, one type 2 small bipolar cell gives output to 10 outer hair cells, or 10 outer hair cells ultimately signal to one type 2 bipolar cell. From my class of synapse, you can recall that this is an example of convergence. So, in the outer hair cells, there is lot of convergence, while in the inner hair cells, there is lot of divergence. Structurally, the most important component of any hair cell, whether it is outer or inner, the structurally most important component is its stereocilia, which are the projections from their apical membrane. Stereo means it has the ability to move, and it is a cilia, and they project from the apical membrane of each hair cell. Each hair cell has about 30 to 150 rod-shaped stereocilia projecting from their apical surfaces. These stereocilia have cores composed of actin-myosin filaments. There are parallel filaments of actin along with various isoforms of myosin. These actin-myosin filaments gives these cilia the ability to move. Now, these stereocilia are not uniform in nature. They vary in diameter as well as in height. Their diameters can vary from 0.2 to 0.8 micrometers, and their heights can vary from 4 to 10 micrometers. Means some stereocilia are taller compared to others, and some stereocilia are stouter compared to others. And within the clump of the processes on each hair cells, there is an orderly structure. Means these stereocilia are not haphazardly ordered. There is an arrangement. And what is that? One extreme stereocilia is the tallest, and the rest are arranged in progressively decreasing height from it. Very fine processes called tip links tie the tip of each stereocilium to the side of its higher or taller neighbor, and at the junction of these connections or tip links are cation channels that are mechanically sensitive. The resting membrane potential of the hair cells is about -60 millivolt. Now, at rest means the stereocilia are straight, they are not bent in any direction. Even though when they are straight, a small but steady leak of depolarizing potassium current flows through the base of the cells to the perilymph. As I have said earlier, the tips and the tops of the hair cells are bathed in endolymph, while the bases are bathed in perilymph. Perilymph is just like extracellular fluid, so it has very low potassium concentration, and so potassium current, a steady leak is occurring always throughout the bases of the hair cells, and it starts pushing the potential towards the positive side. Now, when the shorter stereocilia are pushed towards the taller member, the membrane potential is decreased because the open time of the channels which are linked with the tips are now more, and the membrane potential reaches about minus 50 millivolt, and the hair cells gets depolarized. Now you will ask, why will the stereocilia be moving? Actually, movement of endolymph that occurs due to movement of the basilar membrane, that occurs due to the traveling of the sound waves, is causing all the movement of the stereocilia. Now, as I have said, the potassium ion, which is the most abundant cation in the endolymph, will now enter into the hair cells through the cation channels which are open, and this entry of potassium will cause opening of voltage gated calcium channels which are located in the basal parts of the hair cells. Entry of calcium as well as potassium will cause further depolarization of the hair cells. Opposite thing occurs when the hair cells are pushed in the opposite direction, that is, when the shorter stereocilia is pushed away from the taller one. The cells are hyperpolarized because the cation channels linked with the tip links, they are now less open, and the conductance through these channels decrease. This is a schematic diagram of a hair cell. On the left hand side you can see there is a depolarized hair cell, while on the right hand side is a hyperpolarized one. As I was saying, what has caused the depolarization? The movement of the shorter stereocilia towards the taller one has caused mechanical deformation and opening of cation channels at the tips of the stereocilia. Potassium, which is high in concentration in the endolymph, has entered into the hair cells, causing its depolarization, and ultimately the voltage gated calcium channels has opened, and vesicles from the basal part of the hair cells have been secreting the neurotransmitters, which is causing the depolarization of the afferent axon. While on the right hand side, because the taller stereocilia has moved towards the shorter one, the mechanical deformation has closed the cation channels at the tips of the stereocilia, so no potassium has entered, so no calcium has entered, so no vesicle has been secreted the neurotransmitter. As I have already said, depolarization of the hair cells causes the vesicles in the basal part of the hair cells to release neurotransmitter. The neurotransmitter is glutamate, and this glutamate causes further depolarization of the afferent neurons. Opposite things happen when the hair cells are hyperpolarized, means no glutamate is released and there is no depolarization of the afferent neurons. Now, the rate of action potential firing in the postsynaptic axon is a function of the transmitter output of the presynaptic hair cell. What does that mean? Means the neurotransmitter output of the hair cells determines the rate of action potential firing in the postsynaptic axon. So higher the rate of firing means the neurotransmitter output is also higher. Or in other words, the movement of the stereocilia, to the shorter one towards the taller one and the taller one towards the shorter one. These two determines whether the hair cells are depolarized or hyperpolarized. When it is depolarized, it will release neurotransmitter. When it is hyperpolarized, it won't. So if this to and fro movement occurs quickly, the release of neurotransmitter and stoppage will occur quickly. So the action potentials will occur quickly in the postsynaptic neuron. Why will this occur quickly? If the basilar membrane is moving up and down quickly, the endolymph will move quickly and the stereocilia moves quickly to and fro. Hence, this to and fro movement of the stereocilia determines the rate of action potential firing in the postsynaptic neuron, which in turn will determine our sound perception. When I will teach sound perception, I will further elaborate on it. What happens to the potassium that enters the hair cells? They are recycled. First of all, they enter the supporting cells and they passes on to the further supporting cells by way of leaky tight junctions. So the tight junctions between the hair cells and the supporting cells is tight junction, while the tight junction between the two supporting cells are leaky in nature, through which potassium can go through. Eventually they reach the stria vascularis, which I have talked about when I was discussing the diagram of the organ of Corti. The stria vascularis again secretes the K plus ions back into the endolymph, completing the cycle. Now we come to the most important aspect of sound perception, that is how sound is transduced through the auditory system. What is sound? First of all, it is a perceptual phenomena and is produced by periodic waves of pressure through air or any other media that has the ability to cause periodic movements of its particles. Sound can travel through air, through water, through solids also. Now, these waves of pressure, that is sound, they vary in frequency, amplitude and direction. Means we can perceive how the sound is in terms of its pitch, whether it is a sharp or high pitched sound or a low pitched sound. We can say whether the sound is loud or not so loud and from which direction the sound is coming. Our auditory system is specialized to discriminate between frequency, amplitude and direction of sound. Now, perception of sound involves the conduction of waves, firstly through the external auditory canal that causes vibration of the tympanic membrane. This vibration of the tympanic membrane is transmitted through the ossicular chain of the middle ear cavity into the liquid media of the inner ear, where the hair cells convert them to nerve impulses which is transferred to the auditory cortex for final sensing. Again, perception of sound involves the conduction of the waves through the external auditory canal, causing vibration of the tympanic membrane. This vibration is again transmitted through the ossicular chain to the liquid media of the inner ear, where the hair cells perceive this vibration in the liquid media, convert them to nerve impulses, and they are transferred to the auditory cortex for final sensing. The first step in sound conduction involves the in and out vibrations of the tympanic membrane when the sound wave falls upon it after it traverses the external auditory meatus. This in and out movement of the tympanic membrane functions as a resonator and reproduces the vibrations of the sound source. The vibration of the membrane stops almost immediately when the sound wave stops. The next structure in line are the auditory ossicles. These ossicles together function as a lever system that converts the resonant vibrations of the tympanic membrane into movements of the foot plate of the stapes. As I have already mentioned during my discussion of the functional anatomy, that the foot plate of the stapes overlies the oval window and is attached to the posterior margin of the oval window, just like a hinge door. The resonant vibrations of the tympanic membrane is converted into a swing-like to and fro door-hinged movement of the foot plate of the stapes. So this to and fro door-hinged movement of the foot plate of the stapes is ultimately convened to the perilymph-filled scala vestibuli of the cochlea. The sound waves which are coming from the outside travels through the external auditory meatus, causes in and out resonant movements of the tympanic membrane, which causes movement of the ossicular chain. Ultimately, there is a to and fro hinge door-like movement of the foot plate of the stapes, and this movement is transferred to the perilymph-filled scala vestibuli of the cochlea. Thus, what we learn now is that during conduction of the sound waves from the environment up to the cochlea, they travel from air-filled media of the middle ear to fluid-filled media of the inner ear. So what is the problem in this? Due to a change in the media, the sound waves show the tendency of being reflected away, because the ear and perilymph has different acoustic impedance. That is, one media tries to resist its deformation brought about by pressure changes in the other. This phenomena, where there is acoustic impedance due to change in the media, is also known as an impedance mismatch, and if it had been allowed to persist, mathematically it has been found out that there would have been 97% reduction in the sound energy that is transmitted to the inner ear. However, due to the phenomena of impedance matching within the middle ear and the inner ear, this phenomena is greatly circumvented. Impedance matching. What is it? It is the process by which the acoustic impedance between the air and the fluid is circumvented so that the loss of incident sound energy is not that much. What is the basic mechanism by which the human ear achieves it? Basic mechanism is by increasing the sound pressure of the incident sound waves on the oval window. How does it do so? It does so by two basic mechanisms. Firstly, the tympanic membrane is almost 17 times larger in terms of surface area compared to the oval window. As we have already seen, the in and out movement of the tympanic membrane is transferred to the footplate of the stapes, whose to and fro movement causes the perilymph to move. As the tympanic membrane is much larger compared to the oval window and the footplate of the stapes that covers the oval window, hence the pressure of sound that falls on the tympanic membrane is almost increased to a factor of 17 times when it reaches the oval window. Or in other words, the sound pressure is increased 17 times due to the larger size of the tympanic membrane. Also, the ossicular chain that transfers the resonant movements of the tympanic membrane to the oval window has a mechanical advantage of 1.3. It has been derived mathematically. Both these two combined together increases the sound pressure by about 22 times than it would have happened if these were not present. That is, the incident sound pressure on the tympanic membrane is increased 22 times when it reaches the footplate of the stapes covering the oval window. So whatever reflection of sound is occurring due to acoustic impedance is circumvented by this increased sound pressure. It has been mathematically found that a 27 decibel increase occurs due to the phenomena of impedance matching. The concepts of decibel I will discuss later. I shall be ending this session here. The next session I shall be discussing about the process of sound transduction in the inner ear, the processes by which the pitch, the loudness and the localization of sound is achieved by the human ear. Also, I shall be dealing with the concept of decibels, the concepts of audiometry and applied aspects, mainly deafness. So thank you students for your patient hearing. The third part and the final part, I would say, will be coming up in a few days' time. Thank you.